Welcome back friends, welcome back in another video tutorial from microbiology with Shagnik. In that video tutorial we are going to discuss about the key properties of viral vector. That is going to be the second video lecture on the viral transduction. In the previous video we have discussed about the introductory part of the viral transduction, where I have told you that virus transduction is only used for delivery of those delivery of gene on those cells that are hard to transfect, right? And there we have also discussed about the advantages and disadvantages part. So in the advantage part we have told you that it has a high in vivo transfection efficiency, okay, and the second advantage is that it facilitates a sustained gene expression, okay. On the other hand, it has couple of disadvantages too. For example, the immunogenicity, second cytotoxicity, third the laborious production preparation, and the fourth one is the different packaging capacity, and the number 5 is the different type of infectivity of the viral vector preparation. So along with various kind of advantage, it has coupled of disadvantages too. Then we have discussed about the standard protocol of the recombinant viral vector. That includes first of all the genetical engineering of the virus, then the transfer of the viral vector to a packaging cell line. So we are transferring the viral particle towards a packaging cell line for amplification purpose, and here the transfection is non-viral transfection. Then from that we will purify the amplified viral particle, then we will undergo a titration, then we will transfer the amplified viral particle in a specific cell of interest. That is how a standard protocol of recombinant viral delivery system works. So that was the quite quick recap of the first lecture video. In the second lecture video we are going to discuss about the key properties of the viral vector. So although viral vector each viral vector are used for different purposes, as we have discussed in our previous video that viral vectors are highly specific, but each of the virus vector should have some common properties to be a good viral vector, okay. So the first property that we are going to discuss about is the safety. As the vector's name suggests, that viral vector, right? So viral vector, that means it may be harmful, and in most of the cases we are developing a viral vector from a pathogenic virus, okay. So we have to keep in our mind very carefully that viral vector will be developed in such a way that there will be a minimal risk in the handling of those vectors, okay. And how it can be accomplished? So here we are doing a deletion mutation. This deletion mutation will mutate the gene responsible for the replication of the virus, okay. As a result, it will just capable to act as a delivery boy and deliver your gene of interest into the host cell. That's it. It cannot further facilitate the formation of new active virions that it used to does in case of the normal viral cycle, okay. So another thing I want to make very carefully clear, that we have to keep another thing in our mind, that the system do not contain any kind of helper virus. So what is the helper virus? Helper virus concept is very easy, that virus contain the gene that we have mutated in case of our viral vector, and as a result, if helper virus is present in the system, it can help the viral vector to replicate, and that is why the name helper virus. Okay, so the absence of helper virus is highly preferable, otherwise the virus can develop the active virion. So we have to keep it very carefully in our mind while developing or while performing any kind of experiment. And another important thing that you have to remember in the safety point, that is the chance of insertional mutation. So what is insertional mutation? What is insertional mutagenesis or insertional mutation, what you want to say? So what is the meaning of the term insertional mutagenesis? So insertional mutagenesis means, so when we are infecting the viral vector, there can be a chance that the viral DNA will integrate with the chromosome in such a way that it will inactivate or disrupt the expression of the tumor suppressor gene and activate the oncogene, and as a result the normal cell is converted into a malignant cell. Okay, that is the term insertional mutagenesis we are using, because upon insertion of the viral DNA, the conversion of normal cell into a malignant cell is going on. That means the insertion of the viral DNA facilitates mutation. So we have to highly avoid this. Okay, so 2 points in safety we have learned. One is obviously the inhibition of the replication, and the other one is obviously the insertional mutation prevention. We have to keep two things in our mind very carefully. Now let's discuss about the second point, that is about the low toxicity. The low toxicity means the virus should have low toxicity or should have any kind of effect, should not have any kind of effect that can hamper the physiology of the cell. Because if the virus have the ability to negatively affect the cell physiology, it will promote the immune system to activate. The immune system of the cell will be activated against the virus under such circumstances, and if it happens, it will not be preferable to us, because then the immune systems of the host will act against the virus and try to destroy it. Okay, so that is the idea of low toxicity, that virus should have no detrimental effect towards the physiology of the cell. If the virus have any kind of detrimental effect, it will facilitate the immune response of the host cell to activate, and that will be not a good news for us. Okay, now let's discuss about the stability of the viral vector. So what is the point stability? So stability means the viral vector should have a genetical stability, because remember here we are inserting our gene of interest in the viral genome, right? So if the viral genome does not have any stability in its genetic material, so there will be rapid rearrangement, and that is not preferable to us, because it will facilitate a detrimental effect in the predictability and the reproducibility of the work conducted by the viral vector. So it should be highly avoided. So the virus who do not, in which there is no genetical stability, those kind of viral vectors should be highly avoided. Okay, so now the third point, it should be the fourth point. Okay, fourth point, that is the cell type specificity. So what is the meaning of the cell type specificity? So it is depending on the condition we are experimenting, the requirement of the experiment. That means if our choice is to transfect a specific type of cell, then we will use such kind of virus that have only a receptor for that specific cell. Okay, and if our choice is to transfect a wide range of cells, so we will modify our viral vector in such a way. Okay, and often for specific targeting of the virus to a cell, we are using a process called pseudotyping. That pseudotyping will ensure the transfection of virus towards only a one cell. So that is the idea of cell type specificity. It is totally depending on the requirement of us. If our requirement is to transfect a wide range of cell type, so we can design our viral vector in such a way, and if our goal is only to specifically transfect a one type of cell, then we will use the pseudotyping to develop our viral vector in that way. So that is the idea of cell type specificity. Then the last one is selection. That is quite similar to what we have discussed in case of other vector. So selection criteria means the virus should have a appropriate selection marker that can be a antibiotic resistant gene that will ensure the presence of cells that only contain the viral vector along with the gene of interest. That means that is what we have discussed about the primary selection and secondary selection in case of plasmid. Okay, that means the virus should have a particular antibiotic resistant gene that will select only those cells that have the viral vector along with the gene of interest. Okay, although this point is not applicable for lambda phage vectors, because in lambda phage vector we have discussed that lambda phage vector is naturally selected. Okay, so in that cases, due to the presence of phage headful mechanism, we are not have to bother about the selection. But in other cases, say for example baculoviral based vector, adenoviral based vector, lentiviral based vector, oncoretroviral based vector, we have to keep our attention towards selection also. Okay, that means a presence of a antibiotic resistant gene that will select only those cells that have the viral vector along with our gene of interest. So that is the idea of selection. So these are the various important features that a good viral vector should have. One is safety, where the insertional mutagenesis should be prevented. On the other hand, the replication of the virus should be prevented. Low toxicity, the virus should have a minimal toxicity. Stability, so in case of stability, we should avoid those kind of virus that are not genetically stable. And in case of cell type specificity, this is completely depending on what is our need. If our need is to transfect a wide range of cell types, then we can do by modifying the viral vector in that way. Or if our goal is to transfect only a specific cell type, then we can use the process called pseudotyping to make or develop our viral vector in that way. And the last important one is the selection. Selection will ensure the presence of the viral vector inside the cell, and it is nothing but a antibiotic resistant gene. So that are the various key features of the viral vector that you need to know. So from the next video we are going to discuss about various kind of viral vector systems. So we will start our discussion in next day on adenoviral based vector. Then we will discuss about retroviral, lentivirus and adeno associated virus in the upcoming videos. Okay, so till then, bye bye and thank you for listening to this class.